A very big hello to my wonderful PU2 Warriors to our Brahmastra series. Well, my dear children, this particular series brought a lot of sentems the previous year, and you have to trust me on that. So the previous year when I taught biology and my children went to give the examination, I was like very much tense thinking that how they would, deal with the exam pressure in the examination hall and things like that. But it was not very far that the results came in, and I never ever expected these many number of sentems, to be very honest, because definitely as a teacher I put my efforts to teach you guys, but seeing those children, my students, putting the same efforts, not the same, 10 times more efforts than me, and giving their best and scoring full in biology made me realize one very important thing, that is, if at all someone has the determination, if at all someone is ready to put that hard work into their preparation, they can do whatever they want. And I can guarantee you the same, that if at all you follow each and every video of our Brahmastra series, you will definitely get sentem, my dear children, because it has been planned in that way. Over here, I am going to deal with all the questions from the recent model question paper that was released for 2025-26 from this particular chapter. I would be dealing with questions that has the tag of difficult and average in your question bank, again, the revised question bank of this particular year. Now, the question bank contains the previous year questions, okay, contains the previous year model question paper questions, everything. So you do not worry. Even if you think that I know this chapter, you should still see this particular video, because this would be helping you to settle down everything that you have taught, you have studied, okay, and this will make you score this 11 marks that you can get from the chapter Sexual Reproduction in Flowering Plants. And my dear children, as I said, I guarantee you, being a teacher, I am telling you, I guarantee you sentem marks only on one condition, that is, you stay consistent with this preparation. And if at all you believe me, and if at all you have that trust on me, do not forget to subscribe to the channel for getting all the updates regarding what videos are coming through, whether it's biology, chemistry, physics, mathematics, whatever it is, watch the video, it is for you guys. Okay, and that's all. I'll go ahead. Well, according to the blueprint of this year, that is 2025-26, you get one question from each category, 1 marker, 2 marker, 3 marker, 5 marker. So this particular chapter gives you one question from each category, which means it is important. So if at all you watch this video, probably you would be gaining 11 marks in total, because anyway you would be knowing everything about this chapter because it's there here in this particular video. Okay, and we will start with the model question paper questions for the year 2025-26. I have put down the question number as well, such that you can just give a tick if at all you want. Okay, now those of you who do not have a hard copy of the model question paper, my dear children, you can also put a tick within the soft copy in the computer. You have editing things to do. Okay, you can do that as well if at all you need to do that. Otherwise, it's totally cool. So why I'm telling you to do that is that basically whenever I complete a chapter, I am dealing with model question paper questions, so you get to know that this question is done, this question is done, this question is done, and later on, after a 2 to 3 days gap, probably you can try to do it again, at least the MCQs, and think that, okay, am I able to remember it or not? So revision is also very important. So the first question for model question paper 1, this is also question number 1. So statement 1 says, pollination does not guarantee the transfer of right type of pollen. What do you mean by pollination? Pollination, ma'am, it means that there is a flower, there is an anther over here. From the anther, okay, from the anther, basically the pollen is landing on the stigma of probably some other flower, of the same flower, or another flower of a different plant altogether. It can happen in severe different ways. So pollination does not guarantee the transfer of right type of pollen. Is that true or false? Does it guarantee transfer of right type of pollen? My dear children, this can be plant A or species A. This can be plant B or species B. Still pollination can happen. Still transfer of pollen can happen. A bee does not know where it has to go. It just goes where it has nectar, right? So it will go transfer the pollen. Does it guarantee right type of pollen transfer? No, it does not. So basically this particular statement would be correct. Does not guarantee the transfer of right type of pollen. Statement 1 is absolutely correct. Now statement 2, compatible pollen pistil interaction guarantee double fertilization. First of all, what do you mean by compatible pollen pistil interaction? So you know what? Whenever a pollen lands on a stigma of a female flower, my dear children, what exactly happens is that there is some chemical dialogue happening over here. The stigma talks to the pollen talks to the stigma. It's not like real talking. Basically, there are some chemical messages being transferred. There are some receptors over here. There are some proteins on the surface of the pollen as well. There are a lot of things happening. Now, if at all they match, just like how you have a lock and key, okay, if at all both of them match, then only it opens, right? Similarly over here, if at all the pollen is compatible with the stigma, okay, over here, then only what will happen? The pollen over here will germinate to form a tube. Will form a tube which is known as pollen tube. So it has to be compatible to form this tube. What do you call this? You call this as pollen pistil interaction. You call this as pollen pistil interaction. So compatible pollen is very much important for the formation of pollen tube. But over here what is being said? Compatible pollen pistil interaction guarantee double fertilization. What do you mean by double fertilization? So you know a pollen grain, my dear children, carries 2 male gametes. It carries 2 male gametes. So eventually what has to happen? I'll give you an idea of things. So this is the ovary of the the female reproductive part. This is the style and this is the stigma. In the ovary you have the ovule. Okay, you have something which is known as ovule. Inside the ovule there is one more thing which is known as the embryo sac. And inside the embryo sac you have the most important cell of the female, that is the female gamete. Okay, it has the female gamete. Now, ma'am, there are 2 male gametes, right? So, 2 of them fuses with 1 female gamete? No. So, basically, this tube comes over here, gets inside the ovule, get inside the embryo sac. One of the male gamete fuses with the egg cell out there or the female gamete out there and forms the zygote. Does the fertilization and forms the zygote. Ma'am, you said 1 more male gamete is there? Yes, there is 1 more male gamete. That 1 more male gamete fuses with another cell which is also found inside the embryo sac. It is not the egg cell, it is some another cell which you call as central cell. Now, ma'am, does it form zygote again? No, it does not form zygote. It forms something that is known as endosperm. Ma'am, why there is a need of endosperm? The zygote which is being formed, it needs nourishment. So, endosperm is the one that provides nourishment for that. So, anyways, if at all you see, there is 1 fertilization that took place. There is 1 more fertilization that took place, which is why it is known as double fertilization. The reason I am explaining things to you in detail is that you would be able to solve any question that comes from this particular part of the question. You get it? That's why I'm explaining to you in detail. Now, that is double fertilization. If we come back to the statement over here, compatible pollen pistil interaction guarantees double fertilization. That means it will guarantee the formation of pollen tube until here, and the pollen tube will get inside and the male gamete will transfer and all of this will happen. Does it guarantee that? No, my dear children. Even though it's compatible, pollen tube growth can happen, but it can get stopped as well. It can get stopped, it will be just there, it won't grow as well. There are chances like that. So there is no guarantee for double fertilization to happen. You should understand that, even if it's compatible. If it was incompatible, you do not even think of that, okay? But if it is compatible also, we cannot say that it guarantees double fertilization. Only when the pollen tube enters the embryo sac and transfers the male gamete, we can say yes, there are chances that double fertilization can happen. So can I say statement 1 is correct, but statement 2 which is there, it would be incorrect? Yes, ma'am. So C would be your answer for this particular question. Now, going ahead with the next question, that is a 2-marker question. I told you in the MQP or in the blueprint, one type of question from each category is coming, right? So, next is a 2-marker question. This is question number 21. So, you have to draw a diagram of a mature 2-celled pollen grain. two-celled pollen grain. So listen to me. Over here, this is the actual picture. This is for your reference. I'll draw the picture for your understanding. Now there is one big cell. Okay, this big cell undergoes mitosis. Okay, what happens at the end of mitosis? You need to have two cells. So this cell remains like that. It has some irregular nucleus around it, and there comes another cell which lies in the cytoplasm of this very big cell. So there is another cell. This another cell has a different nucleus as well. So it should have formed two separate cells, but that is not happening. It is actually forming two separate cells, but this cell is in the cytoplasm of the bigger cell. What do you call the bigger cell? Vegetative cell. You can remember very big, V for very big. Okay, now this cell which is there, what do you call that? You call that as generative cell. Why generative? Because, my dear children, the pollen grain which is there, it has to form the gametes. It is not the gamete. It has to form the gametes. So this generative cell is going to generate the gametes for that. How, ma'am? This will undergo one more round of mitosis. Again, it will undergo mitosis, and at the end of it, this generative cell will split into two male gametes. Okay, so ma'am, why do we have this one big vegetative cell out there? This provides nourishment for the generative cell, and also later it provides nourishment for the gametes which are being formed, and there is one more function. I'll be talking about that as well. So anyway, this is a cell, which is why you have a plasma membrane. So you have a plasma membrane. Now apart from that, pollen grain, always remember, has two walls, two very important walls. Okay, so one wall is continuous, does not have any pore. You call this wall as intine. Now, intine is made up of Priyanka Chopra. So you can remember why Priyanka Chopra said is that P for pectin and C for cellulose is just for you to remember. Or you can remember PC. Okay, computer, PC. And that way also you can remember. You might get a question like intine is made up of pectin and something is peptidoglycan. Don't get confused. It is pectin and cellulose. Okay, pectin as well as cellulose. Now going ahead, you have one more layer. Now this layer is a fabulous layer, super important layer. Okay, but you know what? It has some pores in between. Ma'am, why? I'll tell you that. First of all, what do you call this layer as? You call this layer as exine, and it is made up of the most important material over here, which is known as sporopollenin. Ma'am, why do you call it the most important material? Because it can withstand high temperature, alkali, acid, anything. If you put alkali on it, if you put acid on it, it will still be there. The pollen will be still safe. The gametes within it will be still safe because it is made up of the most resistant organic material, which is known as sporopollen. You get a question regarding sporopollen as well always. So please take it into your notice. Now wherever sporopollen is not there, okay, here sporopollen is not there. That means there is a pore there. Okay, there will be a pore. What do you call this pore as? You call this pore as germ pore. I'll write it over here. You call this pore as germ pore. Ma'am, why do you need that? Well, this germ pore helps in germination. Remember, pollen landing on the stigma and then forming a pollen tube. This process is known as germination. So through the germ pore, my dear children, a tube is being formed. A tube is being formed. Now, what helps in the formation of tube? This vegetative cell, remember, it is the one that will be helping in the formation of the tube, which is why the vegetative cell is also known as tube cell. I told you it has one more function. So one function is to nourish the generative cell or to nourish the gametes. The another function is to form the pollen tube as well. Cool. Which is why it is very having a dense cytoplasm and things like that. Now the extra things that you need to notice over here, as I said, first is sporopollen. Second, what is intine made up of? Third, what is the shape of nucleus in a vegetative cell and in a generative cell? If you see the vegetative cell, it is irregularly shaped. The nucleus is what? Irregularly shaped. If at all you see at the generative cell, the nucleus is elongated. It is long. Please remember, you get questions regarding that as well. Now the next type of questions that can come from it is that how many cells are there in total in a pollen grain? Now pollen grain, my dear children, it can have two cells if at all the condition is like this, or if at all you go beyond, the generator cell is also broken down. It undergoes mitosis. In that case, you have 1, you have 2, you have 3 cells. Okay, so a pollen grain can have 2 cells or it can have 3 cells as well. So ma'am, the pollen grain is shed, okay, shed from the anther in a 2-cell stage or a 3-cell stage. Listen to me carefully. 60%, okay, the maximum number of flowering plants, that is known as angiosperms, release the pollen grain in 2-cell stage only. When the pollen lands on the stigma and then the pollination, germination happens, at that particular point of time, it will be breaking into what? 3-cell stage. Until then, it will be 2-celled only. Now, in the rest 40%, it will be in 3-cell stage. This is again a question. So, maximum number is for 2-celled. Minimum 40% is for what? 3-celled. Cool. So, I will be showing you questions regarding it and you will be astonished to find the same concept every single where. Okay. So, let's go ahead. So, before I deal with that, I want to deal with another question. Just give me a quick little second. Yeah. I think it will be this. Okay. So, there is a question in the same MQB, that is Model Question Paper 1 itself. This is question number 42. Okay, over here you are asked, complete the given tabular column with respect to the male and the female gametophyte of the angiosperms for the mentioned character. Male and the female gametophyte. What do you mean by male gametophyte? The one that will be forming the male gametes. What is that? It is, my dear children, the pollen grain. It is the pollen grain. So you are being asked about the male gametophyte or the pollen grain. So over here, mature male gametophyte or the pollen grain. The number of cells present in them. How many? It can be 2 or it can be 3. Now, number of gametes present in them. In my dear children, a pollen grain, how many gametes are there? Always remember, there are only 2 gametes. So, 2 gametes. Now, type of cell division involved in the development of a pollen grain. So, pollen grain develops with the help of something that is known as mitosis. Okay. Something that is known as mitosis. The cell divides into 2. You get vegetative cell and generative cell, all of it, right? Now, ploidy of the cells of. What is the ploidy? That is again a different question. Ploidy means, is it haploid? What is the content of the chromosomes in them? So, my dear children, always remember. So, you have something which is known as microspore mother cell. Okay, it is known as microspore mother cell. They are the ones that will divide and form pollen grains later on. So this cell is a normal cell. You call it microspore mother cell. It is having diploid number of chromosomes. That means double the number of chromosomes. A normal cell has it. Okay. So now this MMC which is there, it undergoes 2 rounds of meiosis. It undergoes 2 rounds of meiosis. In the first round, you get 2 cells, but you know what happens? It gets half. The number of chromosomes become half. It is N and N only. You now call this condition as haploid condition. Okay. Again, I told you there is second round of meiosis. So, meiosis 2 happens. Here also, meiosis 2 happens. You get 4 cells at the end. All of them, they do not get halved again. It will be the same. It will be N, 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 N. They are all haploid in nature. Well, what do you call them? You call them as microspores. They were microspore mother cell, right? So, they are the mothers of the microspores out there. The microspores are grouped like this, 4 of them. You call them microspore tetrad. Later on what happens? Basically, they lose the water between there and they will separate. They will separate and each of them will form pollen grain. Each of them will form pollen grain. So, cell of a pollen grain will have how much number of, or what is the amount of chromosome out there or the DNA content out there? It will be just haploid because it is developed from a microspore itself. So, it has to be what, my dear children? Haploid. So, generator cell has haploid set of number of chromosomes. Similarly, a vegetative cell also has haploid. Why haploid? Because, my dear children, this generator cell will form the gametes. The gametes again will be haploid. You get male gamete and it has to fuse with another female gamete. N and N comes together to form 2N and it will be back again with the same ploidy like a normal cell out there. So, over here, the ploidy will be what? Haploid. Okay, so we have filled in this particular column just with the information of pollen grains, and with that you get half of your marks, right? Now you have female gametophyte, we will come to that as well. Now let's get back to the question out there. So we solved this question, and the next question, which is a 3-marker question, this is question number 28. Now you have to draw a neatly labeled diagram of a monocot embryo. You have to draw a neatly labeled diagram of a monocot embryo. What do you mean by monocot? Monocot means, mono means one, and cot stands for cotyledon. Remember, my dear children, I told you there is zygote and there is endosperm. And I told you endosperm is there for the nourishment of zygote, such that the zygote, which is unicellular, it is made up of single cell, can divide and form an embryo. Now when an embryo is being formed, my dear children, okay, what it does is that it says that I do not need much of the endosperm, which usually helped me. Now I will have my own reserve materials in the form of something that is known as cotyledons. So there are embryos which have 2 cotyledons, that is a dicot plant. Now over here it just has 1 cotyledon. It just has 1 cotyledon in this particular embryo, which is why it is known as monocot. So basically it looks like this. An embryo will have an embryonal axis, something that will give rise to shoot of a plant, that is stem, leaves, everything, and something that will give rise to the root of the plant. Okay, so this part is known as plumule, this part is known as radicle. Now since it is a root, it has to be covered by some protective layer, which is known as what? Your root cap. Now my dear children, I told you it just has 1 cotyledon. So there will be 1 big cotyledon, ma'am? Yes, agreed. So ma'am, so you are saying that endosperm was totally utilized by the embryo and it formed the cotyledon. Yes, it usually does that, but since it is a monocot, it does not completely utilize it because it is just making 1 cotyledon. It's not making 2, right? So some amount of endosperm is still going to persist. So this is the embryonal axis. This is a cotyledon. The only cotyledon which is there, since it is having 1 cotyledon, it pampers it a lot and gives it a name as well, which is why it is known as scutellum. Okay, the cotyledon of a monocot is known as what? Scutellum. So my dear children, monocots are the ones that you cannot break into 2. For example, pea. Chickpea, if you take, okay, chickpea you might have eaten it, right? Kabuli chana you call it. If at all you try, you can break it into 2. That means it is a dicot. You take a wheat. Wheat, can you break into 2? No, my dear children, because it is a monocot. Barley, wheat, rice, all of it is monocot. Okay, always remember, it has just 1 cotyledon and that is scutellum. And my dear children, monocots have some additional things above the plumule. You call it as coleoptile. P over here for plumule. Now similarly, additional layers up over here, which is known as coleorhiza. R over here and R over here. Coleorhiza down. Fine. So this is the basic idea. Embryonal axis, cotyledon, endosperm, coleoptile, and everything out there. Now ma'am, how do we end up in this particular diagram? Because this is a diagram given in an NCERT. So I hope this is clear. If this is clear, I can help you in making that diagram as well. Okay, so just have a look at it. If at all you want to take a screenshot, you can. Well, you will be getting the PDF as well. You just have to join our WhatsApp channel. Okay, now I'll tell you how you can draw this. So basically, make the mouth of a whale. Okay, it looks like this, the top view. Okay, now the whale has 2 hands. One hand goes like this, and there is a small hand over here as well. Now let's make the tail of the whale. So these are the tail, and now we have to join it with this. Done. Now my dear children, make 2 caps. So this is 1 cap and this will be the second cap. 1 cap which is up there, you call it as coleoptile. The second cap which is up there, you call it as what? Plumule. Okay. Now my dear children, this part is nothing but your cotyledon. Okay, this is your cotyledon. So you call this as scutellum. Done. Now over here, you just have to extend these things. So extend it, make it go like that and make it come back again. So extend this, go it like that. Inside within that, you just have to make 1 more rectangle and then bring it back and go to one particular tail, only towards 1 tail. Okay. This also same, you extend it and go towards 1 tail. This region which is out there, you call this as a radicle. Now again you make a cap up over here, you call this as the root cap. Now the last part we know it is what? The coleorhiza. This is the diagram that you have to make. This will give you marks. That's all. I know it might seem difficult, it might not make sense as well. Well, definitely I agree, this picture also does not make sense, but this is there in our NCERT and we have to make it. So coleoptile is one cap, the small cap is plumule. Over here you have radicle, okay, then you have a cap around it, root cap, and then the extra part is known as coleorhiza. Now this is one cotyledon. You know what? One more cotyledon tries to form, but it fails. It is rudimentary, it is not able to form. So this is known as epiblast. It is known as epiblast, the rudimentary cotyledon, the one who tried. Now you can get the same question in a different format as well. And this happened in this year's MQP question paper as well. So draw a neatly labeled diagram of monocot embryo is given over here. In the next MQP, that is model question paper, you could find the same question but now given in a different format, that is, please draw a neatly labeled diagram of a grass embryo. So grass is what, my dear children? It's a monocot itself. So you just have to draw this diagram. Okay, you do not have to draw any other diagram out there. Grass is a monocot. So is barley. So is castor. So is coconut. Okay, so is wheat. Everything out there, they are all monocots. Please remember that. And I'll show you that particular question as well. So I have put the diagram again. This. So this is a 3-marker question again that came in model question paper 2. This is question number 28 where you are asked, draw a label diagram of LS of an embryo of grass. What is this grass? This is a monocot. So it's again the same diagram. Cool. Now we go back again with the question that we have. That is the 5-marker question. One part of it we have solved. Okay, so it has A and B. Always whenever you look at the question, make sure you're reading the entire thing properly. Sometimes we miss the next part of the question, so don't do that. This is question number 42 of MQP1 itself. We have solved one part and let's solve the other part. Now, the mature female gametophyte, and we do not know about it. What is the mature female gametophyte? The one that will be forming gametes in the case of females. What is it known as? It is known as the embryo sac, my dear children. It is known as the embryo sac. And ma'am, how does it look? Look over here. So embryo sac has certain types of cells. Okay, there are 3 cells over there which are known as antipodal cells. Each of them have their own nucleus. Okay, now there are 2 cells right over here, you call them synergid cells. And ma'am, you told embryo sac has the female gamete as well. Yes, now we have the female gamete which is known as egg cell. Remember, synergids and egg cell, they are best friends, which is why you call them egg apparatus. You call it egg apparatus. Now ma'am, now what? Now basically there are 2 nuclei which is lying around there. Ma'am, 2 nuclei, where is the cell? So basically this entire thing that you see is one big cell out there, and what do you call the cell as? You call the cell as central cell. It has 2 nuclei. And my dear children, as I told about microspores, how the microspores develop and how you get the pollen grain which is haploid in nature, because microspores itself were haploid, right? So they gave rise to the haploid pollen grain as well. Similarly over here, all of these cells are haploid. So this is haploid N, N, N. Over here, this is haploid N, N. Egg cell also haploid. But if we talk about the central cell, this nucleus is haploid, another nucleus is also haploid, N and N. Now it becomes what? 2N. So central cell is the diploid one, rest all of it, it is haploid. Please remember. And if you count the number of cells, so we have 1, 2, 3, 4, 5, 6, and this is 7th cell. So we have 7 cell. They are 7-celled, and what about the nucleus? 1, 2, 3, 4, 5, 6, 7, 8. 8 nucleate condition. So they have 7-celled 8 nucleate condition in the embryo sac. Okay, now over here, if we come back to this particular slide and we try to solve it, number of cells present in, how many number of cells are there? Ma'am, there are 7 cells. Okay, number of gametes present, how many gametes are there? Only one gamete, that is the female gamete. Type of cell division involved in the development of. Ma'am, what is the cell division? I do not know, ma'am. So basically, as I told you, just like how you get microspores at the end, similarly here you would be having megaspore mother cell. It will be forming 4 megaspores. It will be forming 4 megaspores. But there is a unique thing about megasporogenesis. So basically, in megasporogenesis, here basically in microsporogenesis, all the 4 of them are going to give rise to pollen grains. But in megasporogenesis, 3 of them are going to degenerate. Only 1, my dear children, only 1 will form an embryo sac. And this is a normal cell that you have, this 1 megaspore which is there. You call this as the functional megaspore. Now this functional megaspore will have 1 nucleus, right? A cell will have 1 nucleus. But you know what? 3 rounds of mitosis happens. 3 rounds of mitosis happens. The nucleus divides. Once it gives rise to 2. Again, you have 4. Again, this gives rise to 2. This gives rise to 2. This also gives rise to 2. This also gives rise to 2. How many nucleus is there? 1, 2, 3, 4, 5, 6, 7, 8. 8 nucleus are there. After how many rounds of division? After 3 rounds of mitosis. After 3 rounds of mitosis, you get what? These 8 nucleuses, and these 8 nucleuses now will move around to different corners, as I showed you earlier over here. Okay, as I showed you over here, all of them will move around and 2 nucleuses will be in the middle. The 2 nucleus will stay in the middle. You get it? So again, what is the division that you need for the formation of embryo sac? It will be again mitosis itself. And remember, 3 rounds of mitosis. Over here, you just need 2 rounds. 1 for forming vegetative and generative, another 1 for again dividing generative to 2 gametes. Okay. Now, ploidy of the cells. We know all the cells are haploid except what? Except the central cell. Except the central cell, which is what? Diploid in nature. And how do you denote diploid? You write 2n. Okay, you will be writing 2n. Now, if we come to this part of the question, so we have filled in all the information right over here. You can read it as well. Okay, the PDF, you will get it. If at all you have not got the PDF, put it down in the comment section that, ma'am, I have not got the PDF, can you help? I'll definitely take that into notice. And my dear children, are you enjoying the session? Do you think it is useful for you? Or do you think it's like a useful question out there? Whatever you think, if at all you love this session, first thing that you have to do is click on the like button. Okay, make sure that it reaches to all the children who really want to top their board examination and any other competitive examination where PU2 is on priority. Okay, so please do that right now. Now, which advantage offered by seeds help the angiosperms to adapt to changing environmental condition? Now, that is a different question, B type of question, B part. Okay, what is the advantage offered by a seed? Okay, to angiosperms that helps the angiosperms of the flowering plants to adapt to different environmental conditions. So basically here you have to talk a lot about the seed. Ma'am, how is a seed formed? I do not know how is a seed formed. It's very simple. Listen to me carefully. Okay, so basically when I say inside the ovule there is embryo sac and there is male gamete and the female gamete diffuse, doesn't it form the zygote? It forms the zygote, right? Now this ovule which is there, where zygote formation has taken place, you will now not call it as an ovule, but rather you will call this as a seed. You will call this as a seed. Now, if you know the structure of the ovule is not alone, it is covered by 2 layers. This is one integument, this is another integument. Now, when it forms the seed, will you now call it as an integument? No. You will now call them as seed coat, and they are also going to get transformed. They will get transformed. It is known as testa and tegument. It is known as testa and tegument. Anyways, the reason why I am telling you is that there will be seed formation and there will be seed coat. Now you are asked, what is the advantage of a seed? Basically, a seed has seed coat which allows it to protect the growing zygote or the growing embryo out there. The seed coat allows the embryo to be protected. That is one very important thing. And apart from that, since the seed coat is covering the whole thing, it allows the seeds to stay dormant as well. What do you mean by dormant? Basically, suppose the seed is being there in one particular situation where there is no favorable condition, there is no water, there is no sunlight, anything. At that particular point of time, if at all you think that this zygote divides to form an embryo forms a plant, do you think that plant can survive? Obviously no. Germination should not happen. So, to prevent it, it enters a stage which is known as dormancy. In dormancy, the metabolic rate, the rate of division, the rate of addition, everything, it stops. It slows down, not stops, it slows down. Okay, that is known as dormancy. So basically, this structure of a seed which has a seed coat and everything allows the seed to stay dormant, also protects the embryo. So seed coat protects the embryo and allows to remain dormant during what? Unfavorable condition. Now whenever there is favorable condition, how does the plant get to, how does the seed get to know? So there is a what do you call, pore over here which is known as micropyle, my dear children. Through this pore, through this small pore, water and everything can enter, and later on it gets to understand that yeah, now it's favorable condition. Now the next thing, what is the advantage of a seed? My dear children, I told you it's not just a zygote that is being formed, it is also the endosperm. It is also the endosperm that is formed. And also, now when the zygote transforms into embryo, the embryo will have its own food in the form of cotyledons as well. So initially you had endosperm, then you have cotyledon in the seed. So food for the embryo is what? Seeds have stored food like endosperm or cotyledon that nourishes the young plant during germination. That is also there. Okay, that is an advantage. Food is also there. Now dispersal. Seeds are very light usually, and they are adapted in a way that they can be spread by wind, water, animals or any other resources as well. So that is also there. So there are three advantages of a seed. First is protection and dormancy. Second is food for the embryo, and then there is dispersal. Now if you do not have, if you are asked 4 points, then please make this as a separate point. Put that seeds allow dormancy as well. Okay, cool. Now that has been done, and now we enter MQP2 questions. Some of them questions we have already solved, as I told you, but let's get ahead. So this is question number 1 again. Statement 1 says, apomixis is the production of seeds from unfertilized ovules. We just studied as of now that basically when an ovule undergoes fertilization, what happens? It will transform into a seed. When an ovule undergoes fertilization, it will transform into a seed. Now this statement is, apomixis is the production of seed from unfertilized ovule. So ma'am, here fertilization is not needed. That is what you are saying, and this process is known as apomixis. Is that what you are saying? Well, yes. So there are some exceptions where the ovule, my dear children, transform into a seed without fertilization happening, and this exception is known as apomixis. This is a process. What exactly happens over here? So usually we had haploid egg cell, right? So it needed haploid male gamete as well for fertilization to form a diploid zygote. Suppose the egg cell over here is already diploid. Somehow the egg cell is already diploid. Do you think you need another gamete out there? No. This egg cell itself can form the zygote. And now since the zygote formation has taken place anyhow, with or without fertilization, now you can call this as a seed. Zygote formation anyway took place, right? Which is why now you will be calling this as what? A seed, my dear children. And here, since there is no fertilization needed, this will be known as apomixis. So apomixis is the production of seeds from unfertilized ovules. Yes, ma'am. Statement 2 says, embryos produced from apomictic seeds are not generally identical to the parent plant. Read it again. Embryos produced from the apomictic seeds are not generally identical to the parent plant. Now I am saying the zygote transformed into embryo, and now this embryo is not going to be the same like the parent plant that it produced, the embryo. Is it true? Wrong. Why? Because the whole genetic makeup is from the female gamete, right? So obviously it will be just similar to the female plant out there, the parent plant out there. So this statement would be wrong. It is written not generally identical. Wrong, because this is a type of asexual reproduction itself. In asexual reproduction, we have studied in amoeba, and amoeba divides into two to give two daughter amoebae. It's like clones that are being formed. You cannot understand what is what. It looks so same. Similarly over here, you are going to get clones in apomixis. So whatever, suppose there's some great character in this female plant out there. It is drought resistant, pest resistant, everything. My dear children, the same features would be transmitted to the embryos as well, which is why apomixis is considered to be good. Now this does not happen naturally in all plants. Okay, I'll tell you the names. Please remember, it happens in grasses, Asteraceae and everything. Grasses and Asteraceae, please remember that it happens in that. You might be given an example of it and then also you might be asked, so you should know. Okay, now going ahead, so statement 1 which is there, it is correct. Statement 2 which is there, it is wrong because it is saying it is not generally identical. So statement 1 is correct, statement 2 is false, you have A option. Cool? Now, going ahead with the next question. Now, as I said, half of the questions we have discussed it before itself because the concepts are repeated. Write the significance of sporopollen. We discussed where is sporopollen? It's in the outside layer which is known as exine. Now, sporopollen is one of the most resistant organic substances, forming the exine of the pollen grains and protecting from high temperature, strong chemicals, UV radiation, microbial attack, everything. Now, because of this extreme durability, pollen grains with sporopollen get preserved as what? Fossils also. They can be preserved as fossils as well. Always remember, it's because of sporopollen. And remember, it is not inorganic but organic substances. So now what I have seen is that since there is not too much to gamble around in biology, they change certain topics, certain words. So instead of putting sporopollen is one of the most resistant organic substances, they put inorganic substances. And you see all the line is same, just that one particular word is wrong and you would not be able to identify that as well because everything else is correct. You get it? So that is how they play with you in assertion reason question. Make sure you're reading everything carefully and make sure you're keeping a note of all this very important terms as well. Okay, so this is MQP2 question. This is question number 21. Now, there's a 3-marker question. We already solved it. Draw a label diagram of LS of embryo of a grass. We know, ma'am, it's just a monocot embryo that is being asked. Now, my dear children, you tell me, are you enjoying the session? Do you think that however time I have utilized over there, it makes sense to you now? Well, it will make more sense to you because there are so many similar questions that you might be seeing. Now, explain the different outbreeding devices developed by the flowering plants. That is something new. So you have to tell the different outbreeding devices developed by the flowering plants. What do you mean by that? Outbreeding means something that prevents self-pollination. I want the plant to outbreed, go breed with some other plants out there of the same species, but I do not want self-pollination to happen. I do not want this to happen. The flower is there, the female and the male part is there, and I do not want fertilization to happen like this. Why? Because this causes something which is known as inbreeding depression. Inbreeding depression. So, ma'am, depression is there in plants as well? Yes, to a certain extent. So, what exactly it means is that, my dear children, suppose this particular plant which was there, okay, it has some very toxic traits such as not being drought resistant, not being pest resistant. It's good for nothing as of now in plant scenario, in plants family, okay. So it is not able to withstand any climatic conditions out there. Every property of it is bad. And that particular condition, if you take the male gamete of the same plant and if you use with the female gamete of the same plant, it's the same character which is going to the embryo as well. Now, do you think the embryo will be able to survive? No. Which is why I'm saying it causes inbreeding depression. It does not allow the embryo to survive itself. Okay, now what are the outbreeding devices that are developed by the flowering plant? That is the next part of the question. So, these are the several outbreeding devices. First thing, time difference. So, pollen release and stigma receptivity are not synchronized. You can put it as no synchronization between pollen release and stigma receptivity as well. I am trying to make it as easy as possible, which is why I put time difference. So, time difference basically I meant to say that suppose the pollen is being released, but the stigma which is there on which it has to land, it is not receptive. It is not receptive. So, in that particular case, my dear children, what will happen? The pollination will not happen and it can prevent self-pollination. Now, spatial separation. Spatial separation means there is some separation in the form of distance, the positioning and everything. So, suppose anther and stigma are positioned differently. Suppose, my dear children, the male part is over here and the female part is over there at the top. Do you think it can easily go at the top? No, it will be very much difficult. So spatial separation is another thing that has been put up by the plants itself. Now self-incompatibility, this is a genetic mechanism. This is genetic mechanism means what? If it is of myself, like the pollen grains are from the same plant, that is me, my dear children, then suppose it lands. Okay, suppose it lands, there will be some genetic mechanism to stop the pollen tube growth out there. This is known as self-incompatibility. You're incompatible. You're making it incompatible. So there are genetic mechanisms that prevent self-pollen from fertilizing the ovules. Now, the fourth one is production of unisexual flowers. Production of unisexual flowers, how does that help, ma'am? Suppose, my dear children, over here I have given you a picture of two plants. What is this plant? Well, this is papaya. Okay, this is papaya. Now, papaya over here you have the female flowers. Female flowers as in the ones that has the female part, the stigma, style and the ovary. Now, I have a whole different flower plant that is a male plant. Okay, with male flowers. So aren't the sexes separate in two different plants? So obviously pollen grains have to come from this plant to this plant. Genetic difference over here as well. You get a new plant with new genetic trait, a mix of the genetic trait. Okay, what do you call these type of plants which has two separate, what do you call, sexes? You call them as dioecious plants. Okay, you call them dioecious plants. So dioecious plants has unisexual flowers. That means only one sex is there in one particular plant. So dioecious plants are the ones, okay, dioecious plants, where is it? Just a second. Yeah, dioecious plants are the one that basically has male plant and female plant separately. This will have unisexual flowers. Example is what? Please remember papaya. This question usually comes. Papaya, 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 papaya. Okay, so male and female flowers on different plants prevents both autogamy as well as gytogamy. This also is a question. In papaya, is autogamy and gytogamy prevented? You should know. What do you mean by autogamy? Autogamy means basically, my dear children, there is a flower from here to here. Things are being transferred. This is known as autogamy within the same flower. Okay, that is anyway prevented because you just have unisexual flowers. Okay, there is only male part or either female part. So autogamy is prevented. What is gytogamy? Gytogamy means this particular flower, there are two flowers on the same plant. Now pollen grains are landing on the stigma of a different flower but on the same plant. That also cannot happen because you just have one sex over here, right? You just have one sex. All the flowers in a dioecious male plant will be just having the male part, not the female part. Then how do you think autogamy and gytogamy can happen? Right? So in dioecious plant, autogamy and gytogamy is totally prevented. Only thing that can happen is xenogamy. Xenogamy means one particular plant, another plant, and you cross it. That is xenogamy, X-shaped. You can remember like that. Okay. Now over here, there is example of monoecious plants as well. What do you mean by monoecious? Monoecious means basically they have sexes like this. So over here, suppose this is a plant. So there can be a flower which can have just the female parts. There can be another flower which can, the male parts. And over here you can have another flower which has the female parts. In that particular case, okay, autogamy is prevented because it is not happening within the same flower. But gytogamy, if at all you see, my dear children, gytogamy can still happen. Gytogamy can still happen. What do you call this? These are again unisexual flowers in a bisexual plant because you have two sexes in one particular plant. This condition is known as monoecious condition where male and the female flowers are on the same plant. If you talk about flowers, they are unisexual, but you have two type of flowers over here. Okay, you have both male and female, which is why autogamy is prevented but gytogamy is not prevented, but still a good approach. My dear children. Remember, this is a bisexual plant. In this papaya, it was a unisexual plant with unisexual flowers. Okay, so monoecious condition is like this. Example is castor and maize. Again, you can get a question. In castor and maize, autogamy happens, gytogamy does not happen. Is it true or false? Or basically, you can get option. What can happen, what cannot happen. So you should know autogamy is nowhere going to happen, but gytogamy can definitely happen. Okay. Fine, xenogamy cannot happen? Yes, there is a different plant just like this, then they can do cross-fertilizing as well. That is also possible. But autogamy is prevented for sure. So this is unisexual flowers. Please remember all the examples and things like that. I have explained it to you. Now, this is a picture actually of castor, I think. Yeah, this was castor. This is castor, you have heard of castor oil, right? So they have bisexual, this is a bisexual plant having unisexual flowers. Cool, now let's go ahead. So this is a 5-marker question. If at all you write your answer like this, you will get the whole 5 marks. This is question number 35 of MQP 2. Now we go ahead with MQP 3, model question paper 3, my dear children, where you have question number 1. So we are almost around 48 minutes, something like 48 minutes by now. Please do not lose your trust. Let me complete. So after the MQPs are completed, we will do more of MCQs, and then we will come to 2-marker, 3-marker, 5-marker questions. It won't take too much of your time, probably 1 hour 30 minutes or 1 hour 40 minutes. But in this particular time, if at all you are able to complete the whole chapter, then do you need to worry about the time? Obviously no, right? So even in the absence of pollinating agents, seed setting is assured in. Seed setting is assured in. So I do not need pollinating agents. Without pollination, ma'am, how pollinating agents, how can pollination happen and things like that happen? Basically, it can happen. Suppose the flower is like this. It is closed flower having the male and the female part within this particular closed flower itself. In that case, my dear children, autogamy can surely happen, right? So ma'am, are there flowers like that which has closed flowers? Yes. What do you call them? You call them clistogamous flowers. Okay, clisto, CL for closed, you can remember like that. Now my dear children, there will be some open flowers as well in the same plant. In the same plant, there can be some open flowers as well. These are known as chasmogamous flower. They are open, which is why they are known as chasmogamous flowers. So ma'am, here anyway autogamy is going to happen. Nothing else can happen, right? Yes, sure. And ma'am, what is the second feature? Ma'am, here I'll definitely get seed setting. That means seed is going to be formed definitely. Definitely seed is going to be formed. What is the next character, ma'am? Do I need pollinating agents? No pollinating agents required. No pollinating agents required. These things. Now, genetic variation, ma'am, is there genetic variation? No genetic variation as well. Why? Because male and female of the same plant itself. So, no much, right? No much genetic variation. Why did I give you these 4 points? Because you might get a 2-marker question saying what are the features of clistogamous flowers. So, please write all of this. Okay. Now, chasmogamous open flowers are also there in the same plant. Now, what are the examples of the plant having these type of flowers? You have 3 examples: Viola, Oxalis, Commelina. Viola, Oxalis, Commelina. So you can remember cow, but over here there is no W, there is V. Okay, Viola, Oxalis, Commelina, and over here, even in the absence of pollinating agents, seed setting is assured in what? Cucurbita, papaya, maize, Commelina. The answer would be Commelina. Why? Because Commelina, my dear children, will be having clistogamous flowers. And usually remember, clistogamous flowers are more towards the root within the soil, or might be just above the soil. Okay, might be just above the soil. I think I have a picture of it as well. Yes, I have the picture. So these are the open flowers that you have, and these are the closed flowers that you can see. It's mostly towards the root as well, but the top also sometimes it's there. Okay. So it's towards the root of the rhizome region, sometimes it can be there, but the chasmogamous flowers are at the top itself. This is Commelina. Okay, okay ma'am. Now, this is a 2-marker question which is not being mentioned as 2-marker, but please remember it's a 2-marker question. And over here it goes like this. Question number 21. Give reasons. Micropyle remains as a small pore in the seed coat of the seed. I told you. So there is integuments that is covering the seed, right? There is integuments that covers the whole seed, but there is a region that is left aside that is known as micropyle region. Always remember, everything important happens towards the micropyle region. Where are synergids and egg? In the micropyle region. Where does fertilization take place? Through the micropyle region. Okay, and then where does the megaspore mother cell start its division to form the megaspores? In the micropyle region. Everything in the micropyle region. Where are the antipodal cells? In the opposite side, that is chalazal region. Okay, chalazal region. So anyways, micropyle is a pore which is found in the ovule. Now when this ovule transforms into a seed, it still persists there. Why do you think it persists there? So that the transfer of moisture and everything can happen. Let's read it. The micropyle of the ovule persists even after fertilization and becomes a tiny pore on the seed coat. You might have seen a tiny pore, okay? It helps the entry of water and oxygen during seed germination so that it remains which is why it remains unchanged as a small opening. So whenever there is water and things like that, it enters. Also gaseous exchange takes place over here itself, such that it gives a signal that yes, seed germination can happen. You need not be dormant anymore. Okay, so that is the use of micropyle. Now apple and cashew are not called true fruits. Why? So ma'am, what is a true fruit first of all? Then only we can say why they are not called as a true fruit. So over here, my dear children, we studied there is ovary, inside that there is ovule, right? An ovule transforms into a seed. What happens to the ovary? The ovary transforms into a fruit, my dear children. It transforms into a fruit. Inside the fruit, you have the seed, right? So it's usually the ovary that transforms into a fruit in true fruits, okay? But in some cases, what happens is that the ovary is sitting on the thalamus, right? Now this thalamus can also participate in the formation of a fruit. Along with the ovary, the thalamus can also participate in the formation of a fruit. In that case, can I call it a true fruit now? Because it's not just the ovary, it is also something else. No, it won't be known as true fruit, it will be known as false fruit. Example is apple, cashew and also strawberry. Apple, cashew, strawberry are examples of this. They are all false fruit. And why are they known as false fruit? You should know that. I told you, true fruits develop only from the ovary after fertilization. But in apple and cashew, the edible part develops from the floral parts other than the ovary. Floral parts, these are the parts of the flower. So please write the exact thing. It develops from the floral parts other than the ovary, like the thalamus in apple and the pedicel or the accessory tissue in the cashew. So this is pedicel over here. In cashew, the pedicel also is taking part over here. In the case of apple, the thalamus is taking part. Hence, they are known as false fruit or accessory fruits, not true fruits. So over here, you have a picture in your NCERT as well. If at all you have not seen the picture in your NCERT where they have given you picture of an apple saying that this is the thalamus and things like that, you are not reading your NCERT properly. Please make sure you open your NCERT and see the diagram very, very nicely. It's just about the parthenocarpy thing, okay? So what exactly happens over here is that, so if you see this part, okay, I'm not talking about this part, I'm talking about this part. This is from the thalamus. Now if you see this part, my dear children, this is your ovary. Your ovary, and that inside that you have the seed. So ovary is inside, the bigger part is the thalamus. Now the ovary, they have mentioned endocarp, mesocarp, this carp, that carp, things like that. So what usually happens is that in the case of an ovary, right? So suppose I have mango. In mango, you might have eaten a mango, right? And in mango, it looks something like this. So the outermost layer, so this is the fruit and this is your seed. So I'm leaving that. I'm leaving the seed. I'm talking about the ovary. Ovary has a wall which is known as pericarp. The wall of the ovary is known as pericarp. Now the pericarp is divided into three layers. The outermost layer is known as exocarp. Exocarp. Okay. Now the thing that we eat, the fleshy part, is known as mesocarp. Okay, and the thing that we throw, which has the seed in it, remember the seed will have its own seed coat. I'm talking about the thicker layer which is inside. It is known as the endocarp. It is known as the endocarp. So over here, you do not have a distinction like this. Rather, everything is inside. So you would have your endocarp, the thick portion which you might have seen in the apple just beside the seed, that will be endocarp. And then you would be having your mesocarp to a bit extent, and then there will be an exocarp. It won't be clearly visible, but it's there. And the other part, this particular part, will be known as thalamus. Make sure you're going through it. Again, you can get questions related to that as well. Cool? Now, so that is also done. So a 2-marker question is done. You get your 2 marks with that. Apple, thalamus, and in cashew, pedicel. Okay? Next question is, draw a neatly labeled diagram of a typical anatropous ovule. Anatropous ovule means ulta ovule. So now, my dear children, first of all, we will see what is the structure of an actual ovule, and then we will see structure of an anatropous ovule. It will be easy then. So first of all, we have to make the ovary. It looks like this. You have your ovule. I told the ovule has two layers. So basically, ovule feels very cold, which is why it has two sweaters. And it has unbuttoned the sweaters. This particular region is known as micropyle. And over here, these are your integuments. Please remember, you have to mention like inner integument and outer integument as well. Now it's inside the ovary, right? This is the wall of the ovary. What do you call this space as of the ovary? You call this as locule. Okay, this is known as locule. That is the open space. I'll write it properly so that you remember. Locule. Fine. The ovarian cavity. Now both of them cannot stay separated like that. It cannot be in air. So basically there is someone to nourish the ovule in general, and this part is known as placenta. It is known as placenta. Placenta is connected to the ovule with the help of one very important thing, which is known as funicle. Okay, it is known as funicle. And the point of attachment which is there, it is known as hilum. So funicle says hi to the ovule with the help of hilum. So it is the point of attachment. This is the general thing. So if this is the micropyle region, the opposite end will be known as the chalazal end. It will be known as the chalazal end. Now, this is ovule. You also call the ovule as megasporangium, because, my dear children, a lot of things happens over here. Megasporogenesis, everything happens over here. Now, if you go within the ovule, you have certain cells. You call these cells as nucellus. All of it together, you call them as nucellus. This is just a normal cell, a diploid cell out there. There will be one cell which forms the megaspore mother cell. Only one cell undergoes division. In the case of in the case of anther, the microsporangium has multiple MMC and it forms multiple pollen grains as well. But over here, there is only one MMC. Okay, please remember that as well. So this is general structure. Now, how do I make anatropous ovule, ma'am? So this particular structure which is there, the placenta and the funicle as well as the hilum, this would not be here. Rather, they would be somewhere here. So this is the placenta, you have the funicle and you have the hilum. Okay, you have the funicle and you have the hilum, my dear children. This would be what? Anatropous ovule. Make it bigger and it will be all clear. So what did they do? They shifted the thing. So funicle is right over here and there is hilum, the point of attachment. This picture is not that clear, but there is a picture in your NCERT. You can refer to that as well. Just make it ulta. That's what I am saying. Do not use too much of your brain. You might you can end up stressing, so don't do that. And the opposite end will be always chalazal end. So please don't think that wherever placenta is there, that will be chalazal end. No. Opposite end of the micropyle region will be chalazal end. That's all. Okay. This is a 3-marker question. So diagrams are coming in 3-marker questions, if at all you have noticed that, in MQP1 and MQP2. Now this is a 5-marker question. Okay, and this is from MQP3, question number 35. Answer the following. Double fertilization is a unique event taking place in flowering plants. Briefly explain. Okay. Arrange the following terms in the correct developmental sequence. This. So basically over here you would be asked about the sequence of microsporogenesis. Can we do this first? It's easier, right? In microsporogenesis, I told you what happens, my dear children. You have microspore mother cell. Okay, this microspore mother cell divides into what? Undergoes meiosis, forms haploid cells, again undergoes meiosis, forms 4 microspores. 4 microspores. What do you call them? You call them microspore tetrad. Tetrad means 4 of them. Now all of them are going to separate out and you get microspores. And these microspores, the separate ones, give rise to what? The pollen grains. So ma'am, where does the MMC come from? So basically inside the anther, that is the male part is together known as stamen. Stamen is made up of anther and filament. Inside the anther you have a tissue which is known as sporogenous tissue. Okay, this is known as Sporogenous tissue. Inside that, all of the cells, my dear children, are what? Are MMCs. Are MMCs. So if at all you want to arrange this, first comes sporogenous tissue, ranking 1. And then comes pollen mother cell or microspore mother cell, ranking 2. And then comes what? Then comes microspore tetrad, ranking 3. From microspore tetrad, you get the pollen grains, that is ranking 4. And the male gametes, then the pollen grains, my dear children, what do you get? You have the male gametes. So basically, this will be 5. Cool. And now we go to the first part of the question. Double fertilization, a unique event taking place in the flowering plants. Explain. How does that happen? So see this picture carefully and I'll explain it to you. So, yes. I would need to go back a bit with this particular diagram because I think it will be easily understood by you here. Okay. Okay. So this is your egg cell, right? What exactly happens is that, my dear children, listen to me. The pollen tube comes over here and the synergids which is there, it is having something which is known as filiform apparatus. The filiform apparatus is something that will guide the pollen tube into what? The embryo sac. It will guide the pollen tube inside the embryo sac. Now the pollen tube is guided inside. One of the synergids is going to get degenerated. The male gamete is transferring, that's why one of the synergids is going to get degenerated. And the egg cell and one male gamete is going to fuse. So this is N, this is also N. Both of them fuse to form diploid what? Diploid 2N or diploid what? Zygote. That is clear. This is first fertilization. Now there is one more male gamete. One more male gamete comes. This male gamete is having haploid. This is a male gamete. And this is having haploid number of chromosomes. The two polar nuclei, they are all haploid, N, N. So how many nucleus in total? 1, 2, 3. So we call this fusion as triple fusion. And you get something which is known as triploid. Okay, you get triploid primary endosperm cell. What do you get? You get triploid primary endosperm cell. Now this is a cell. It has to divide to form a tissue, that is the actual endosperm. So as of now, what do we call it? Triploid primary endosperm cell, which has a triploid primary endosperm nucleus. It has primary endosperm nucleus as well. This is known as triple fusion. Why? Because 3 nuclei are fusing. What do you call this type of fusion? The first fertilization that took place. It is known as syngamy. It will be known as syngamy. Since fertilization is happening twice, you call this as double fertilization. Now, the endosperm is the one, this primary endosperm cell is the one that develops first because it has to form the whole endosperm layer, and then this endosperm layer is the one that will be nourishing the zygote to form the embryo, which is why endosperm formation precedes what? The embryo development. Precedes the embryo development or the formation of embryo. Now we'll come back over here. So in flowering plants, 2 male gametes enter the embryo sac through the pollen tube. One male gamete fuses with the egg cell to form a diploid zygote. This is known as syngamy. The second male gamete fuses with the 2 polar nuclei in the central cell to form the triploid primary endosperm nucleus, triple fusion. Well, this primary endosperm nucleus is of what? The primary endosperm cell as well. Please write that. Primary endosperm cell. Now, because 2 fusions occur in the same embryo sac, it is known as double fertilization, a process unique to what? Angiosperms. It is not found in any other type of plants. You have different type of plants. One is flowering, one is non-flowering, such as gymnosperms and things like that. It's not happening in them. Okay. Now the second part of the question is where you had to arrange. We have already arranged it, so you need to get worried. If at all you can, please make a diagram as well to show, because this is a 3-marker question and this might be a 2-marker question. It's a 5-marker question in total, right? So make sure you are making the diagram and explaining. You might get some additional marks out there. And please write your answer in points. Now, MQP questions are done. Model question paper questions are done from this particular chapter. We are going to go ahead with the revised question bank questions, PYQs and other MQP questions of the previous year. So all of it is combined in your question bank, and I have given you numbers like that. You need not worry about anything. Okay, so this is question bank question number 2. I have taken the questions which have been tagged like average, difficult and things like that, because easier ones probably we would be doing in some live sessions as well towards the examination, but as of now I think you need help over here. Okay, so select the correct sequence of anther wall layers. Select the correct sequence of anther wall layers. What is a wall layer of an anther? My dear children, please remember, if the anther looks like this, if I cut it from the top, just a second. So usually it looks like this. This is the filament and this is the anther. Suppose I'm cutting the anther like this and looking from the top. How does it appear to me? That is the question. So it looks like this. So it has two lobes. This is one lobe and this is second lobe. And each lobe, my dear children, has two theca. This is one theca, second theca, third theca and fourth theca. Okay. Now over here, in one corner, you will have the microsporangium, or in one corner you have the microsporangium that is being there. Okay, and inside that you will be having so many, microspore mother cell, each of them giving rise to the pollen grains. Now I am looking at only one. And how these things appear over here? Let's see that. So my dear children, the innermost thing that you have, oh my god, okay, fine, it's okay. I'm just making one side, okay? The innermost thing that you have over here, which is made up of so many cells, right there, is known as sporogenous tissue. It is known as sporogenous tissue. Ma'am, all the cells of the sporogenous tissues are microspore mother cells? Yes. It is made up of what? Microspore mother cells. Now, there should be someone to provide nourishment to this, my dear children. There should be someone to provide nourishment to the growing pollen grains later on, and also these cells which has to divide, my dear children. What do you call this layer as? You call this layer as tapetum. This is a wall layer of anther. This is the wall layer of anther. Okay, sporogenous tissue is a tissue. Now you're talking about layers. Tapetum is a layer. Now tapetum is unique because it is made up of cells which are binucleate. Binucleate means basically one cell might have two nucleus. Why? Because the nuclear division took place, but the cell division did not take place. Cytokinesis didn't take place, nuclear division took place. And also one more feature, they will have dense cytoplasm. They will have dense cytoplasm as well. So tapetum is binucleate and they have dense cytoplasm. Okay. Now you talk about the other layers. The outermost layer right over here is known as epidermis. It is known as epidermis. Now inside the epidermis you have another layer which is known as endothecium. Endothecium. Now in between endothecium and tapetum you have another layer which is known as middle layer. Ma'am, three of them, aren't they very much special? I have read somewhere. Yes, they are special because they will be the one that will be helping in dehiscence of anther. That means for the breakdown of anther and to release all these pollen grains, these three layers are going to play the major role. So they help in dehiscence of anther and also protection. So please remember the flow like this. You might have heard of ENT doctors, ear, nose, throat doctors. Similarly, here it is EMT. Okay, it's EEMT, epidermis, endothecium, middle layer and tapetum. And these are the features of all of them. Tapetum is not involved in dehiscence of anther, please remember. It's just there for nourishment of the sporogenous tissue cells of the sporogenous tissue and also the pollen grains. This can be multiple questions. Okay, there can be multiple questions arising from the same concept, which is why I'm stressing on it. So select the correct sequence of anther wall layers: epidermis, middle layer, endothecium. Wrong. Epidermis, endothecium, middle layer, tapetum. Yes. So your answer for this question becomes what? B. This is a picture you can refer to. Okay. You might be asked to draw this as well. Make sure you practice the diagram. Now next question, select the correct sequence of events in microsporogenesis. We have studied so many times. So sporogenous tissue, then microspore mother cell, then microspore tetrad, then microspore. So first option itself is correct. You have tissue, you have cells in them. These cells, they undergo division to form tetrad, and then all of them, they move away and they form microspores. So this statement itself is correct, but please, whenever you are in an examination situation, read out all the options. Sometimes the first option might be just there to confuse you guys. So you will not read the other option, you will give it a tick, but the other options might have some other correct answer. Over here, it's not there, but I request you to please have that practice. Okay? So here I have explained the microspore mother cell undergoes meiosis once and then meiosis 2, all of them form microspore tetrad. Each of them, they will separate to form unique microspore, which later give rise to the pollen grains. Now 15th question, this is question number 12 of your question bank of part A, okay. From the statements given below, choose the option that is true for a typical female gametophyte of an flowering plant. You have to tell what is true for a typical female gametophyte. Female gametophyte means what, my dear children? Embryo sac. Embryo sac. Now we have studied about embryo sac. We know its division. You have to tell me. Remember, there was one functional megaspore, all the three degenerated. This one functional megaspore underwent 3 mitotic division. The nucleus divided. Was it immediately covered by cells? No. At the end, you get 8 nucleuses and then only they get covered by what? Cell wall and things like that. Remember, I taught you. So it is 8 nucleate and 7 celled at maturity. Is it true? Does it have 8 nucleuses? Yes. Is it 7 celled? Yes, because central cell has 2 nuclei. It is free nuclear during development. Is it free nuclear? Free nuclear means immediately it is not covered by cell wall. Is that true? That is also true, ma'am. Now it is situated inside the integument but outside the nucellus. Inside the integument, embryo sac which is there, my dear children. So you had the ovule. Okay, you had the ovule. We had made a diagram of this, right? Let me come back to this. So here MMC is there. It forms the embryo sac later on. Later on, this will be the embryo sac. Now, suppose this is the embryo sac. In that particular case, this embryo sac is within the nucellus and also within the integuments. Within the nucellus, because this is the cells of the nucellus usually degenerate. Sometimes it may persist as well, remember. And outside that, you have the integuments. Now, we'll come back to this question. Where is this question? Okay, so it is situated inside the integument but outside the nucellus? Wrong. It is also situated inside the nucellus. Now it has an egg apparatus situated at the chalazal end. I told you everything important, micropylar end, not the chalazal end. Egg apparatus has the female gamete. Female gamete super important, not at the chalazal end. So basically, can I say A and B are the only thing that are correct? Yes, ma'am. So A and B is correct, which is why your answer would be C. Now the next question which is there, why I have done this? Okay, so make you understand the free nuclear division. So one mitosis happens, you get two nuclei. Again mitosis happens, you get 4 nuclei. Again mitosis happens, you get 8 nuclei. Now 3 of them arranged there after formation of cell wall. 3 of them arranged there, 2 of them arranged over here, egg is there, and the 2 polar nuclei arranged over there. Okay? Now 16th question. Question number 13 of your question bank, part A. Identify the correct sequence of arrangement of cells in a mature embryo sac micropylar end. So usually if we do not see micropylar end, chalazal end, we just start with the antipodal, this and that and things like. You're asked about the micropylar end. So if at all you see the micropylar end, my dear children, right over here. This is the micropylar end. I told you egg apparatus in the micropylar end. First thing that you have is synergids, then the egg cell, then the central cell, and then the antipodals. Okay, so your answer for this question would be what? First, synergids. So I have 2 options with first synergids. Let me cancel these 2 options. Use the elimination method to be quick. Next, after synergids, you have central cell? No, you have egg cell. So central cell, egg cells, and synergids, egg cell, central cell, and antipodals. This will be your answer. A is your answer. Now, my dear children, look at your speed. Aren't you fast? Why? Because we know the concepts, and I always tell you this. Please understand the concept. It's going to be super easy. I have discussed about this, so no need. Okay. Again, question number 19th. So statement 1 says, in Viola, both autogamy and geitonogamy is prevented. Viola oxalis commuliana. In Viola, do you think both autogamy and geitonogamy is prevented? Autogamy will definitely happen in clistogamous flowers. In chasmogamous flowers, the open flowers, autogamy can happen, geitonogamy can happen, anything can happen. So both autogamy and geitonogamy is prevented would be a wrong statement, so this is wrong. Now statement number 2 says, geitonogamy is similar to autogamy since pollen grains come from the same plant. So my dear children, the usual question comes, differentiate between geitonogamy and xenogamy. This came the previous year and all, okay. If at all you know autogamy, geitonogamy, xenogamy, you can easily put down the differences. Autogamy means what? Same flower, anther to the stigma, the same flower, genetically same. This is known as self-pollination. Geitonogamy and xenogamy, both of them are cross-pollination. But which one is true cross-pollination? Which one is true cross-pollination? This one. Why? Because here 2 plants are involved, 2 flowers are involved, and from here to here pollination is happening. Genetically different type of plants you get. But over here in geitonogamy, 2 flowers are involved, but of the same plant. So genetically, do you think any variations will be there? No. So can I say geitonogamy is similar to autogamy since pollen grains come from the same plant? Yes. Genetically, yes. Genetically, it is similar to what? Autogamy since pollen grains come. That's the thing. Okay, now they have not mentioned genetically, but still you can give this as a correct statement itself. Okay, this can be given as a correct statement itself. Is similar to autogamy since pollen grains come from the same plant is what they have written. So other statements that everything is correct, which is why we can say statement 2 would be correct, but statement 1 is false. So statement 1 is correct. No, statement 1 is incorrect, but statement 2 is correct. This is what we can put down. Okay. I hope you're getting this. Please know the differences. This is genetically same, genetically different. Both of them are cross-pollination itself. Say it happens between 2 flowers of the same plant. It happens between 2 plants itself altogether. Next question. So here I have explained. So this is autogamy where things happen here itself. Geitonogamy like this and xenogamy like that. Okay. Now this question super important, question number 18. This was question bank question number 35. This picture is usually not looked upon, okay, it's there in your NCERT, but children tend to ignore it altogether. So what is this? The following picture is an example for, Mom, I do not know what is this, looks like a drum or things like that. It's a flower first of all. And this flower, my dear children, has multiple carpels. Carpels means what? Carpel, pistil is the same thing. That is the female reproductive part, stigma style ovary. So it has multiple carpels. So it is multicarpellary condition that you see. Okay. There is one more thing that you need to know. These carpels that is there, they are all fused. They are all fused. What does that mean, ma'am? So basically all of them will have just one ovary. This condition is known as syncarpous condition. This is known as syncarpous condition. So there are multiple carpels out there, but all the carpels are fused. They have one ovary, which is why it is known as syncarpous condition. So multicarpellary syncarpous gynoecium. Gynoecium means what? The female reproductive part. Over here, multicarpellary syncarpous androecium is given. That will be wrong. So make sure you're reading the options carefully out there. So this is your answer. What is this particular plant, by the way? It's the plant that gives you the drug opium, okay, heroin, morphine, all these things. So it is known as papaver. This is what? Papaver. This is given in your NCERT just above megasporangium. You can go through that, okay, where ovule and things is given. Okay, now there is, so this is the actual thing, how it actually looks. So you have multiple carpels and all of them are fused. And my dear children, there is one more example given in NCERT that is about Michelia. Michelia. Michelia is what do you call? Champak. It smells so good, okay. In Malayalam you say chembagam. Many Malayalis up there. So basically this in Michelia, what exactly happens is that again you have multicarpellary condition. Multiple carpels are there, but these carpels, my dear children, they are not fused. That means they have the separate ovary out there. Or say the separate ovule out there. Everything is separate out there. Okay, so this condition is known as apocarpous condition. Apocarpous. So that is syncarpous, this is apocarpous. In papaver, it is syncarpous. Here it is apocarpous. Again given in your NCERT. Michelia is like a girl's name, right? Michelle. You can remember like that. Now, my dear children, we are going to enter into what? 2 marker, 3 marker, 5 marker questions. Okay, I've included only a few because most of the concepts are being already taught over here when I discussed about all of it. You just have to listen patiently to me. So question bank part B, question number 4 is this 19th question. Complete the tabular column given below with respect to the male gametophyte and male gametophyte of the angiosperms. This is something that we have already done. I told you we get questions regarding that. The cells of the male gametophyte. There are two cells. One is what? Vegetative. The other one is what? Generative. Now, the shape of the nucleus. I told you it's important. The shape of vegetative is what? Think about it. Think for a second. The shape of vegetative, the bigger cell was what? It was irregularly shaped. Whereas of the generative cell, it will be elongated. Okay, it will be elongated. So that is your answer. There is no too much of drama out over here. It's very easy question, a direct question. If at all you know everything about pollen grain, it becomes a lot more easy. And also remember, what's the diameter of a pollen grain? It's 25 to 50 nu meter or micrometers. Okay, 25 to 50 nu meter or micrometers. Please remember the diameter as well. Okay, now 2024-25 model question paper question is this. Okay, differentiate between pericarp and perisperm. It's not there in the question bank questions. It's there towards the model question paper, the previous year. Pericarp and perisperm. What is the difference between both of them? Pericarp, do you remember? I told you pericarp is what? The walls of the ovary. Pericarp is divided into exocarp, mesocarp, and endocarp. What is perisperm, ma'am? I do not know anything. Is it something related to the male gametes, because that is what you call as sperm? No. So basically we spoke about the nucellus still persisting, right? The nucellus which is there, it still persists. I told you somewhere. Whenever it still persists, for the nourishment of the growing embryo later on, my dear children, this persistent nucellus is known as perisperm. It's very rare, but it can happen. It is known as perisperm. Where do you see it? In two type of plants. You say B square. One B for black pepper and another B for beet. Another B for beet. So black pepper and beet, my dear children, they have persistent nucellus for the nourishment of embryo. You will call this as perisperm. So for nourishment of embryo, you have endosperm, you have cotyledons, and sometime nucellus also persists back, which you call as perisperm. How do you frame your answer? You frame your answer like this. The wall of the ovary develops into the wall of the fruit, and this is known as pericarp. Epicarp is the outer layer, mesocarp is the middle layer, and endocarp is the inner layer. It is the hard layer. Okay, endocarp is the hard layer. Middle layer is usually fleshy. In some, it might not be fleshy as well. It might be dry also. Now, perisperm is a persistent nucellus, okay, and it is found in black pepper and beet. Done. Cool, it's a 2-marker question. You get your 2 marks with that. Now, distinguish between albuminous and non-albuminous seeds. Distinguish between albuminous and non-albuminous seeds. What do you mean by that? Albuminous means here the endosperm is persisting. Non-albuminous means what? Endosperm is used up. I told you monocots have scutellum and also endosperm. So examples of albuminous seeds are what? Monocot. What are the examples of monocot? Grass, okay, maize, wheat, castor, coconut, all of them are examples of albuminous seeds. Non-albuminous, endosperm is used up. Why? Because, my dear children, over here they are forming 2 cotyledons. Example, dicots. In dicots, my dear children, the picture image is very, very simple. If at all you need to draw the diagram of dicots, it's super easy. I'll tell you why. Because over here again, you have the plumule, you have the radicle out there. This is the plumule, this is the radicle, right? Now make a cotyledon that lies over here. Similarly, make another cotyledon that lies over here. This is second cotyledon. This is what? This is your plumule. This is what? Your radicle. Radicle will be having root cap. It does not have coleorhiza. Always remember. This region which is above the embryonal axis, okay, above the axis of attachment. This is known as epicotyl region. In the epicotyl, you have the plumule. This region below is known as hypocotyl. Anyway, they have two cotyledons out there. Here, endosperm is totally used up because you want to form the cotyledons. Now, what are dicots? The one that you can break into two. Green peas, chickpeas, whatever you can remember which you can break into two, they are all dicots. Don't think like coconut I can break into two if I throw. No, if at all you look at its embryo, you will get to know. The coconut which is there, it's the ovary and everything out there, the wall layers. Inside that, the embryo also forms. You might have not seen it. If at all you have not, please Google it once. Coconut embryo, you will be able to see it. It looks like a round ball. Okay. So yes, that is a thing, but if at all you want the little difference, it goes like this. So in albuminous seeds, it retains some endosperm even after seed maturation. Over here, endosperm is completely used up during embryo development. Here, provides nutrition to the embryo from the stored endosperm. Here, nutrition is stored inside cotyledons, not in the endosperm. Example, wheat, maize, barley. Here, example would be what? Bean, pea, groundnut, etc. Okay? Simple. Cool? Let's go ahead. Are you? Fine. Now it's a 3-marker question. I've given the answer as such. So this is part C, question number 15. Fertilization is not an obligatory event for fruit formation in certain plants. Justify the statement. So fertilization is not needed for fruit formation. So until now we have studied fertilization is not necessary for seed formation. Example was apomixis. You studied about Asteraceae, grasses and things like that, right? Now you are saying it's not necessary for fruit formation as well. Do you think so? Yes, my dear children. So suppose over here I have the ovary, inside that you have the ovule. Inside the ovule, fertilization didn't take place. There is no formation of zygote. Still the ovary is forming the fruit. Can that happen, ma'am? Yes, it can happen. It happens because of the effect of some hormones. Some hormones makes the cell active and makes the ovary turn into a fruit. Now in that case, since fertilization has not taken place anyways, the seed formation, do you think it will be there? No, there won't be any seed formation. Here apomixis is also not happening. Don't think like that. Apomixis can happen. No, nothing is happening. Think like that. Since there is no seed formation, fruit is there, there is no seed. Can I call it seedless fruit? Yes. What do you call this process of formation of a fruit without fertilization? You call this as parthenocarpy. You call this as parthenocarpy, and the basic example of this is what? Banana. Seedless. So in most plants, fruits develop only after fertilization when the ovary enlarges to form the fruit. However, in some plants, fruit can develop without fertilization. This process is known as parthenocarpy. Parthenocarpic fruits are seedless because no embryo or seed formation occurs over here. Such fruits can occur naturally, example banana, or can be induced artificially by applying growth hormones such as auxins and gibberellins. So I can induce it as well. How? By putting the growth hormones such as auxins and gibberellins. Now therefore, fertilization is not essential for fruit formation in these plants as it can develop in the absence of fusion of gametes. So they are seedless, always remember that, and explain your answer properly line by line giving the examples. Okay, which is why I have put your answer over here. Now we have next question, which is again a 3-marker question. This is question number 17, part C. So I took the questions from the part which has not been covered at all so that you get an understanding the whole chapter is done. Okay, so why are some seeds referred to as apomictic seeds? Why? My dear children, they are produced without the fertilization. Okay, fertilization process is not happening out there. Okay, the diploid egg which is there, egg cell is there, which it is converting into, or it is developing into what? A zygote which is already diploid. You do not need fusion of gametes. So some seeds are called apomictic seeds because they are found without fertilization or without fusion of gametes. The embryo develops without meiosis and syngamy, producing seeds genetically identical to the parent plant. Now mention its advantages to a farmer who uses them. What will be its advantage to a farmer who uses them? So these are the advantages of apomictic seeds. You can get a separate questions on that as well. So hybrid vigor is maintained. What do you mean by that? Farmers can reuse hybrid seeds every year without loss of yield or quality since no segregation occurs. So basically what is happening here, the female plant is enough for producing the new generation out there. So I've got a seed which is exactly like that. A seed again will produce an embryo out there, a seed which is giving rise to a whole plant. Now this plant which is there again will be producing its own zygote without fertilization. So basically whatever feature is there in this particular seed is getting transmitted. There is no sexual reproduction happening anywhere. You do not need a partner. There is no loss of any quality or yield. So hybrid vigor, whatever hybrid nature was there over here, whatever important features were there over here, it is not mixed because it does not rely on sexual mode of reproduction. Next thing, cost effective. Farmers do not need to buy hybrid seeds every year. Yeah, do they need to buy? No, they can get the seeds from this particular plant itself. Do they need to buy? No, because I have all the features. So usually when it happens that you take a plant, you take another plant, they fuse together and they form another plant. So you have the hybrid feature. Now this plant relies on sexual pollination methods, right? So from other pollen it comes. Suppose this pollen is not that great, aren't you losing the good trait out over here, right? Because now there is a mixing of some other traits and it might not be that great, but over here you do not need all that. Now, uniformity in crops. All plants grown from apoptotic seeds are genetically uniform, ensuring uniform growth and profitable performance as well. So all this or predictable performance. So basically these are the things that you can write and this can give you marks. Hybrid vigor is maintained, cost effective and uniformity in crops. Suppose I want to grow in large numbers, I can easily grow with the help of apoptotic seeds. Cool. Now, this is 24th question, part D question. This is part D, question number 3. Plan an experiment and prepare a flow chart of the steps that you would follow to ensure that the seeds are formed only from desired sets of pollen grains. I have to plan an experiment or a flow chart in which I have to make seeds. I have to make seeds from desired pollen grains itself. Suppose I have plant that has superb quality and I want the same features of a plant over here. What do I do? I have another plant also which has desired feature. I don't mix them. First of all, mixing them will be known as hybridization. Okay, mixing them will be known as hybridization. And since I'm doing it artificially, it's me who is doing it, I'll call this artificial hybridization. So name the type of experiment that you carried out. It is artificial hybridization. Now I write the importance of such experiments. Basically, I can grow my type of plants, the hybrid varieties of plants which has the unique profitable features that I need. That is the importance of such experiments. Now how do you do the experiment? That is again an important thing. So my dear children, first of all, suppose it's a bisexual flower. What do you mean by bisexual flower, ma'am? So suppose it has male part as well as the female part. In that case, I would have to remove the male part because I do not want the pollen grains to land over here. What do you call this process as? You call this process as emasculation. I am removing the masculine features. Emasculation is the first thing. Okay. Now, basically what I am going to do, there are chances that pollen grains from some other unwanted plants can also land over here. So after I remove this, I have to cover this particular thing. It is known as bagging. This is known as bagging. And while I do bagging, I prevent other pollen grains from landing and I also wait for the stigma to be receptive. The stigma to be receptive. Now once I feel the stigma receptive, what do? I open the bag. I do not need to write like. I open the bag and I put the pollen grains of my desired plants. Okay, I put pollen grains of my desired plants. Pollen grains are shed. Now after that, what will I do? Again, pollen grains can come, different pollen grains can come. So I cannot leave it like that. Again, I would have to what? I would have to rebag the whole thing. So the smooth pollen pistil interaction can happen and everything. So this is known as rebagging. Now suppose, my dear children, over here you do not have bisexual flower, you have unisexual flower. That means only one sex is there, the female probably will be there. Do you need emasculation? No, their emasculation is not needed. So my dear children, this is how it goes. You have emasculation, you have bagging, and as I told you, shed the pollen grains and then you do rebagging, all of it. So let's see. To ensure the ovules of selected plants are fertilized only by desired pollen grains, we have to do this. Select parents. Choose desired female parent to bear seeds and the male parent source of desired pollen. Emasculation. In the bud stage of bisexual flower, carefully remove the anthers of the female parent with forceps before dehiscence. Before the pollen anther dehisces, we have to take that away. Now bagging. Immediately cover the emasculated flowers with a butter paper polythene bag to prevent unwanted pollens from reaching the stigma. Pollen collection. Collect the mature pollen from the selected male partner when they enter the hysis. Artificial pollination. Open the bag, dust the desired pollen grains on the receptive stigma of the emasculated flower. Rebagging and tagging. Cover again and tag the flower with details of classmate and date. Done. Seed formation. Allow fertilization and collect the mature seeds formed. That's all. If the female parent has unisexual flowers, emasculation is not needed, as I explained to you earlier. Cool. Now, name the experiment. It is artificial hybridization or controlled cross-pollination. Importance of such experiments to prevent contamination by unwanted pollen and help in producing hybrids with desired characteristics. So, this is one point, this is another point. Such as higher yield, disease resistant, better quality, which are essential for crop improvement program. Now, I'll tell you, the previous year the question came as, farmer used crop improvement program to produce a desired plant. What was this method? So children got confused because whenever you see PU2 chapters, you have biotechnology, applications of biotechnology as well, where you see genetically modified plants as well. So you think that is crop improvement program. Remember, you are being asked about artificial hybridization. Read the question carefully and please write the answers accordingly. Okay, I hope that is done. Now, question number 25. Let me see how many questions are there. More. Okay, this is the last question, my dear children. You are free after that. Picture related to pollination is given below. Answer the questions that follows. Will this pollination confirm fertilization? Justify your answer with suitable reasons. So, I have not included the picture over here. I'm so sorry. So, the picture looked like this, where, my dear children, there was a flower. And not just that, there was honeybee. I do not know how to make a honeybee, but I can try. So this was the picture. Okay, this was the picture like. So you have to tell, picture related to the pollination is given below. Answer the questions. Will this pollination confirm fertilization? Will any pollination confirm fertilization? As I already told you, no. No. It can be incompatible, it can be compatible. If it's compatible, it does not guarantee fertilization. You have to justify your answers with suitable reasons. How do you do that? Pollination only brings pollen grains to the stigma. It is just a transfer process. Fertilization occurs if the pollen is compatible, germinates on the stigma and forms a pollen tube that reaches the ovule. So it's not just compatibility, but it has to germinate, it has to form the pollen tube, it has to reach the ovule and transfer the gametes as well. Now, if pollen grain is incompatible, will not germinate and fertilization will not happen. So, please talk about pollination, what is pollination and what is fertilization. Say that they are individual, independent things. Okay, not independence, they are individual processes out there. So, you have to explain your answer like that. Now, what are the floral rewards provided by the plants to the insects to revisits? So, what are the rewards given by the plants for the insects to revisits, my dear children? They produce nectar. They produce nectar. They also produce pollen grains. So it's not like all the pollen grains are made to travel through and it helps in pollination. Some of the pollen grains are taken by the organism as well, because these pollens are very nutritious. So they are taken up, and while doing that same process, some of the pollen grains attach to their body as well, and that's how it's getting transmitted also. So what are the floral rewards? One is nectar, another is pollen, and there is one more thing, that is shelter. Shelter. You might have seen in yucca and moth. So yucca is a flower and moth is an insect. So basically they show obligate mutualism. Basically they help each other very much. They cannot live without each other. So moth helps in transfer of the pollen of the yucca plant, and yucca, my dear children, provides a safe space for moth to lay its egg inside the ovary actually. So that's how it works. So there's a shelter thing that is there. Apart from that, you have one more example of Amorphophallus. There is the biggest flower out there, Amorphophallus. In that also you provide safe space for the insect pollinators for resting, for laying their eggs as well. So Amorphophallus and yucca and moth are examples for shelter. Please remember that. Go revisit your NCERT just to understand that. Now, mention the characteristics of flower in this pollination other than the floral rewards. So, what are the other features of the flowers, my dear children? They have to be large, they have to be colorful, they have to be fragrant to attract what? Insects. So, large and colorful can be one point. Fragrant flowers can be another point. Now, the sticky or spiny pollen grains, so they can attach to what? An insect's body. The pollen grains should be very sticky, otherwise they will not be attaching to the insect's body. If it is not attaching, it cannot be transported as well. Now the next thing, stigma is sticky to capture pollen by the insects. So that is also there. The stigma has to be sticky such that the pollen grains, when it comes, it can easily trap it up. Okay. Now foul smelling flowers in plants pollinated by flies and beetles. So flies and beetles, they do not need good fragrance, they need foul smell. So that is there. Similarly, you can be asked the question related to wind pollinated flowers. What are the features of wind pollinated flowers? You should know the features. The pollen grain, they are what? They are non-sticky. If they are sticky, can they transfer through wind? No. So the pollen grains, my dear children, non-sticky. Now the pollen grains, do you think they should be light? Yes, they should be light such that it can be easily transmitted. So again, it should be light. Now my dear children, what about the stamen? What about the stamen? Stamen should be exposed, well exposed stamen so that wind can carry the pollen grain. So well exposed stamen. Apart from that, what do we have? What about the pistil? The pistil which is there, my dear children, it should be feathery, or the stigma which is there, it should be feathery such that it can trap. So, just a second. Stigma, feathery. All these points are super important. Example of wind pollinated, always remember you have two examples. One is corncob and the other one is grasses. Corncob and grasses are the example. Grasses, do not forget, it happens here in wind pollination itself. Okay, and that's all. Apart from that, I don't think there is anything else. Cool. So, yeah, and also the flowers which are there, they come together to form an inflorescence that happens over here as well, and also they would be having just one ovule per ovary. Why? Because usually plants can have multiple ovules, but here there is only one ovule per ovary. This is because wind pollination can be rare as well. Again, it can occur rarely, so you don't want to lose your energy in making multiple ovules and wasting it, right? So that is also a thing. So please read your NCERT, it's very much important. Similarly, water pollination, example of Vallisneria. Some homeworks for you guys, for you to do. Vallisneria, Zostera. So these are the examples. One is surface level pollination, one is submerged pollination. Zostera is also known as seagrass. What are the features of the pollen grains? They will be ribbon shaped, they will have mucilaginous covering and things like that. Water lily, water hyacinth do not show water pollination. You should know that. Okay, and there is polyembryony in apomixis. That is also a topic that is not touched. Okay, polyembryony in apomixis, my dear children, what happens is that I told you sometimes the nucellus persist. The nucellus persist, right? And this is your embryo sac. Suppose the nucellus gets into your embryo sac, it can form an embryo. Here also an embryo is formed without fertilization. But here the nucleus cells are involved, and it's not just one embryo but multiple embryo. This condition where multiple embryos are being formed. I'm not saying the formation of formation of embryo without fertilization is known as polyembryony. I am saying the formation of multiple embryos is known as polyembryony. And you see this in citrus fruits. You see this in citrus fruits such as mango, orange, etc. So this is also an example of apomixis, because here the nucleus cells are the ones giving rise to the embryo, but here no fertilization is happening, just like how it happens in apomixis, right? So apomixis can happen because of diploid egg, or it can happen because of this reason as well. This type where multiple embryos are being formed, you call this as polyembryony. You find in citrus plants such as mango as well as orange and things like that. Done. In wind pollination, grass is an example, and with that we wind up. Thank you so much for giving me your time. As I said, I assure that this video is going to be helping you really well. Wishing you all the best. If you think that this video helped you, definitely let me know in the comment section, and do not forget to hit that subscribe button as well. Bye bye children, take care.